Let me get out of this. Whoa, where'd all this stuff come from? Well, sorry about that, guys. I was just trying to get my voice warmed up, taking a little break in between videos. I thought Hocus Pocus would be kind of a good thing to listen to, since sometimes everybody thinks this is Hocus Pocus. But what we're going to be doing today is we're going to learn about how we make protein. And the first step to making protein is making messenger RNA. So that's what we're going to get into today. So we're going to go into gene transcription and RNA modification. And we're going to delve into that and find out what exactly is this all about. So we have a central dogma here that actually was created by Watson and Crick. They came up and they said that we feel like this is what's going on here based upon our understanding of the system, that you had a gene that could replicate itself, and then that gene would express a messenger RNA, and then from the messenger RNA that could be translated into a polypeptide. And so this is referred to as a central dogma of systems. And the polypeptide, which is encoded by the gene, basically you can have, it can be just a simple single-stranded polypeptide, which is a functional protein, or you could be a protein that is actually multi-subunits of polypeptides that come together to bring about a particular function. Now, only one strand of the DNA molecule is actually transcribed into the molecule of RNA. So when this is opened up, one of the strands is going to be the template, and the other one, we refer to that one as the non-coding strand. And so that is how we go about identifying these things. And the interesting thing about this is that because of that arrangement, we could actually have genes that are overlapping each other. And we do find that in nature, especially amongst the prokaryotes, there's a lot of overlapping. And in some cases, in humans, we have some overlapping genes as well. So DNA replication obviously makes copies of DNA, transmitted from cell to parent, from parent to offspring. Chromosomal DNA, as we said before, that stores all the information. The thing to remember, and this is one thing to always catch here, is that DNA controls everything that we're seeing here. It's controlling all the steps in one way or fashion, and it's sending out its molecules from it to help to do that kind of control. So no matter what's going on inside the cell, it is the information stored in the DNA that is controlling all of it. Transcription is going to be producing an RNA copy of the DNA. Now, if you remember, I said earlier, the RNA doesn't stay around very long. It has a tendency to be destroyed very easily, and there's a good reason for that, because if the DNA is kicking out RNA all the time, well, we got to dispose of the RNA. We got to use it, get rid of it, because otherwise we have a buildup of that material in the cell, which could lead to various types of diseases. So that has to be used. And then finally, the message of this type of RNA, which is going to be used to be translated into polypeptide, it's referred to as a messenger RNA because it's providing the message to generate that particular protein. It has to be translated, and we call this translation, where you basically produce a polypeptide using that information. And then this polypeptide becomes a functional protein. It becomes part of a functional protein, or it acts as a functional protein. So let's go through this introduction of the concept of a gene first. Overall a gene is made up of three basic units. You have a region in front of the start site of transcription, which is represented by this bent arrow. This is upstream from that particular start site. We refer to that as the upstream of the gene. That basically is a regulatory region. The purpose of this promoter is to promote the transcription. If you think in terms of human concepts, if you go to a concert like playing Hocus Pocus, if you go there, they're going to set it up in a way to where they're going to have an individual who promotes the concert. And so if you think of the concert, the actual production of the concert itself as the expression of the gene, that's the concept of the promoter. They bring together the musicians, they bring together the vendors, bring it all together to produce the concert. So the promoter's purpose is to bring all the proteins necessary together to produce a messenger RNA transcript. Now the gene, as you can see, it has the promoter as the start site of transcription. This is the RNA coding sequence represented by the green. Now you have to have a conclusion, so that means you've got to terminate this thing. So there has to be a region of the DNA that actually terminates. This is the way you're being presented here. This is a general way of presenting a gene in these box formats here, like this. It's just an easier way. And the way to think of this is the actual, this would be the one strand, and this would be the other strand up here, and this will all be built into the system. Now notice here you have downstream regions of genes, and we always talk in those senses because outside of this is just a basic unit of a gene. Outside of this are going to be other portions of the DNA that's going to help to be volume control on this gene. And we call those things like enhancers or silencers. So transcription means that we've got to bring in an RNA polymerase into the right location to get transcription. You have to make a copy. That's what transcription means. The DNA will continue to store the information, but we have to make a copy from that. To do that, you have to have an RNA polymerase that's going to interact with this. And as I said before, the purpose of the promoter is to get this molecule at the exact location necessary to start transcription. Now, synthesis of the RNA does the same thing as in DNA. DNA synthesis, if you remember, is 5' to 3'. Well, RNA synthesis, biosynthesis, is also 5' to 3'. And you have that same nucleophilic attack that occurs on this particular phosphorus of the incoming ribonucleotide triphosphate. And so then once that occurs, you have a 5' to 3', so you have a phosphodiester backbone, and you have chain growth will continue to grow. And if this is using the template DNA, and notice how this is going this direction, so it's 3' to 5' this way, and this one here is 5' to 3' this direction. So they're anti-parallel even at this case. So let me give you this animation of RNA biosynthesis.
Sequences of DNA nucleotides in genes contain the instructions for making proteins. The process of protein synthesis consists of two main steps. First, DNA is transcribed into messenger RNA. This step is called transcription. Next, the messenger RNA leaves the nucleus and attaches to a ribosome, where the RNA is translated into a polypeptide. During transcription, only the DNA strand in the 3' to 5' direction is copied into RNA. This strand is called the template strand. The other DNA strand is referred to as the non-template strand. Transcription begins at the 3' end of the template strand. An enzyme called RNA polymerase binds to the DNA and unwinds a short segment of the double helix next to a gene. The RNA polymerase synthesizes an RNA strand by selecting the precursor nucleotide that can form a complementary base pair with the nucleotide on the template strand. As in DNA replication, the polymerase then catalyzes the formation of a phosphodiester bond between the 3' end of the new RNA strand and the 5' phosphate of the RNA precursor base. The formation of the bond results in the release of two phosphates from the precursor. This process is repeated as the RNA polymerase moves along the template in the 3' to 5' direction. When the gene has been transcribed, the polymerase releases the completed RNA and detaches from the DNA. Notice that the final RNA molecule is identical to the 5' to 3' non-template strand of DNA, except that the thymine in DNA is replaced by uracil in RNA.
That's a nice little video to kind of give you an overview of that particular biosynthetic process. So a key concept to remember that the DNA-based sequences define the beginning and the end of gene and regulate the level of RNA synthesis. Very important that you understand that as we move forward in our understanding of gene expression. Another important concept is that proteins must recognize and act on the DNA for transcription to occur. So proteins have to initiate the whole concept here. So overall process of gene expression, basically by which the information within a gene is used to produce a functional product, is in concert with environmental factors, is determined by the trait, common organization of a bacterial gene, or excuse me, the trait of the organism, all that. Now this again is the common structure that we see in a bacterial gene, and it can also be reflected in eukaryotic genes. So if we take a little bit closer look at this, on the regulatory sequence, which is going to be the promoter, right along in here, that's the area that's going to be interacting with the RNA polymerase. And then of course you have the coding, and then you have the terminator, and you get transcription. And from that, you now get a messenger RNA. Now the messenger RNA, we'll find out in eukaryotes, is going to be modified somewhat, but in either case, bacteria or the eukaryotes, there has to be a location up here at the 5' end of the messenger RNA that is going to be where the ribosome is going to bind to. So there is even a regulation at that particular point for ribosome binding site, because it has to figure out where the start codon, the beginning of the actual polypeptide, is located in this messenger RNA. Now we'll go into more detail about that in the next chapter. But let's kind of focus on a little bit of this. So you have a start codon in the RNA sequence, so that's going to be AUG, which is the standard one that you know about. And then the actual ribosome will move over to here. It doesn't move from here to here to here. It moves over 3 nucleotides on the messenger RNA, then reads that as a codon for 1 amino acid, moves again, moves again, moves again. These are called frames, all right? And for this to work, you have to have a start codon in frame with a stop codon to stop the actual production of the amino acid. All right, so that means that there has to be something very specific there in place to work in this situation. Now, gene expression basically requires the base sequences to do this. So the DNA strand that actually is transcribed is turned into template, as I mentioned before, and the RNA transcript is going to be complementary to that template strand, meaning that this is going 3' to 5', 3' to 5', it will bind to it in that particular fashion. The non-template strand, as pointed out in that video, is actually a copy of that with just the thymine being replaced by the uracil. So with that, they're pretty much the same on that particular one. Now, you're about to watch an interesting video here, and this video, the reason I like showing this video is primarily because what happens here is a lot of people, even scientists, get confused about how they start this particular transcription. Our concept is that the transcription factor comes in and binds to that particular location. That's the start site of transcription. Well, in reality, it's kind of wrong, because what happens here is these factors that identify the start site of transcription and all this, that bring in a gene that's going to be expressed. There's factors that bind to the DNA. And if you remember, I told you that these proteins will bind to this, and when they bind to it, they act like this is almost like it's traveling along here on its own. And what's going on here is that this is going to be the alpha helix that wraps around here. This particular part of what we call a transcription factor protein binds, and it binds in general to this phosphodiester backbone. And when it does that, it can then move along just like a train, going along until it picks up the sequence it's interested in binding to start transcription. So this is what you're about to watch, and this is very important to understanding how this whole thing is identified. So here we go.
By controlling which genes are active, a cell can take on special characteristics. Muscle cells and neurons have the same DNA but perform different functions because they use different sets of genes. A key challenge in genetics research is to understand how transcription factors find the correct DNA sequences to control genes. In the nucleus, DNA is condensed into a DNA-protein complex called chromatin. In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a